right, what we want to talk about now is a little bit about the genetics of the immunoglobulins. And the reason that we incorporate a part on the immunogenetics, or the genetics of the immunoglobulin molecules, is because the genetics really has explained a lot of the phenomenology. I've been telling you a lot of things. I've been telling you things that B cells have on their surface, both IgM and IgD, but yet the specificity of those, that IgM and IgD is the same. I told you that there's this thing called allelic exclusion, but how does all that happen? And many of these questions really were answered from an understanding of what's going on at the level of the genes for the immunoglobulin molecules. And so many of the things that we've talked about will be explained by the genetics. And so we're going to cover the genetics of the immunoglobulins. And let me just sort of give you a little historical perspective for the immunoglobulin genetic story. When all this sequencing was going on, where people were sequencing the heavy chains and the light chains, one thing became very clear from these studies. The same constant region could be associated with many variable regions. So, for example, you could find IgG antibodies that had a lot of different specificities. All right? So the same constant region that determined a gamma 1 or a gamma 2 or a gamma 3 could be associated with a lot of different antibody specificities. But the sequence studies also showed that the same, if it'll let me, the same variable region could actually be associated with many different constant regions. So, for example, you could find an IgM and an IgG molecule that had the same idiotype. And the idiotype, remember, is a variable region marker, so it meant that they had the same variable region. So here you have the same variable region on two different classes of immunoglobulin. So the question can be asked, well, how can you have a situation like that? How can you have the same constant region associated with a lot of variable regions, or the same variable region associated with a lot of different constant regions? And in an attempt to answer that question, the immunologists suggested at the time that maybe what's happening is that you have two genes that encode the immunoglobulin molecules, a variable region gene and a constant region gene. And you could then just sort of mix and match which genes you're going to put together. You could take this same variable region and put it with a bunch of constant regions, or vice versa. So they suggest, well, there's two genes coding for this protein. Well, obviously it would be easy to explain how you get these results with that. But the problem was, at the time that this suggestion was made, and we're talking about this is going on in the late 60s, early 70s, probably before all of you were born. But that's the time that roughly late 60s, early 70s. The problem is that our understanding or our concept of gene at that time was one gene, one polypeptide. And here these immunologists are saying two genes, one polypeptide. Didn't make any sense. Okay, nobody wanted to accept it. So what happened was that people said, well, no, all right. We got to have two genes. That's the easiest explanation. But then what happens? So each gene makes their protein, because now we got to obey the rule: one gene, one polypeptide. Okay. So now we make two proteins and we stick the proteins together somehow. Somehow you then form a peptide bond between the variable region protein and the constant region protein, and that would do it. Okay. So people then looked for evidence, that the two proteins were coming together. Nobody could find any evidence. In fact, there were some really exquisite experiments done to show that that doesn't happen. There is no fusion at the protein level. So then people say, well, okay, let's make the fusion at the RNA level. So you have two genes, they make two messages, we link the messages together, and then we translate it into one protein. So then they looked for evidence for that. Nobody could find it, all right? And in fact, the evidence said absolutely not, that doesn't happen. It wasn't until the mid-70s that the first evidence came out that something was happening at the DNA level. That in fact there are two genes they are not separated. They happen to be on the same chromosome, but they're not the same gene. They're separated on the chromosome, And some rearrangement occurs at the DNA level. And in fact, we know now that, in fact, the immunoglobulins are coded for by 3 gene families. One gene family encodes the kappa chains, kappa light chains. Another gene family encodes the lambda light chains. And the third gene family encodes the heavy chain genes. And all of these gene families are actually on separate chromosomes. All right, so we have distinct groups of genes that encode these 3 different classes. And what we want to do is then to look at these gene families and look at how they're expressed. So what we're going to do is we're going to look at the way the genes are organized in the germline in a cell that is not differentiated in any way to be a B cell. So this may be a stem cell, or actually it could be any cell. It could be a kidney cell or a liver cell, whatever, anything that is not a B cell. We're going to look at the structure, the organization of those genes, and then we're going to look to see what happens when a B cell starts undergoing B cell differentiation. What happens to those genes? All right? So we'll start with the lambda chain, light chains. This is the gene organization in the germline, in the undifferentiated cells. In the case of lambda, there is actually one constant region gene for every subtype of lambda. So we have 4 constant region genes in this gene family. The one codes for the lambda 1, lambda 2, lambda 3, and lambda 4. And by the way, in all of these in subsequent figures, the boxes are going to represent the exons, the coding sequences. The thin lines in between them are introns. This is the intervening sequences of DNA. All right, so we have there's one exon for each type or subtype of light chain, of lambda light chain. Now, quite a ways away from the constant region genes, and that's why I put a dotted line in here. We're talking about, this is a huge piece of DNA actually, but quite a ways away, you find the variable regions. And there are multiple variable regions. In the case of lambda, there's about 30 different variable region genes that encode lambda variable regions. And each of these genes has this. There's the main exon that codes for the bulk of the gene. Then there's a short intron in here, and then there's a short sequence labeled L that stands for leader region, and we'll see the importance of the leader region shortly. But each one of these variable regions has a leader region associated with it. There's one additional coding sequence in here, which is indicated here in white, and upstream of each of the constant region genes, there's this other J gene segment. And we typically don't refer to it as a gene. We usually just call it a gene segment because it really only codes for just a few amino acids. It's just, 5 or 6 amino acids encoding. So it's just a piece of a gene, basically. So the upstream of each of these, we have a J gene segment. And it's unfortunate, the nomenclature. Don't get confused. That J has nothing to do with the J chain that you find in IgM or anything. Okay, so it's totally different. It's just, the J stands for joining. Okay? All right, so that's the organization of the lambda light chain genes in the germline. What about kappa? Kappas look like this. You have a single kappa heavy constant region gene. There's only one type of kappa chain. There's only one kappa constant region gene. You also have multiple variable region genes separated quite a distance on the chromosome from the kappa gene, from the C-kappa gene. Right? And each of these variable regions have a leader region associated with them. And in the case of kappa, there's about 300, roughly in that ballpark. Those are the estimates, the number of genes. And you also have J gene segments. But in the case of kappa, you have multiple J gene segments upstream of the C-kappa gene. Right? So that's the way the gene looks in its germline configuration. What we want to do now is to look at what happens when a cell in the bone marrow, and when a precursor of a B cell, a stem cell in the bone marrow, commits to becoming a B cell. and undergoes B-cell differentiation. What happens to these genes? And we'll follow the example with the kappa. The same is going to be true of the lambda, so I'll just take you through with kappa. So here's our germline organization with kappa. All right? So what happens? What happens is that there's a DNA rearrangement. A recombination event occurs between one of these 300 variable region genes and one of these 4 or 5 J gene segments. A rearrangement occurs. In our example here, a rearrangement is going to occur between V kappa 2 and J kappa 4. It should be a 4. All right? So between J kappa 4 and V kappa 2. This is a recombination event that occurs. And what happens is all this DNA in between here gets deleted and is removed, so that we now bring V kappa 2 in our example now immediately adjacent to J kappa 4. Now, it is a, quotes, random event as to which V gene is used and which J gene segment is used. And the reason I put random in quotes is because there does seem to be some bias for choosing those variable region genes that are closest to the J regions. So in other words, this gene here has a greater probability of recombining than this gene over here. But eventually, in the B cell population, there's going to be a B cell that will recombine every one of these things to one of the J gene segments. So there's one B cell is going to take J2 next to V2 next to J1. Another B cell may take V2 next to J2. Some B cells are going to take V1 next to J1, and so on. All possible combinations will eventually be expressed, although there does seem to be some preference. The number of cells that use this one to recombine is a little bit higher, okay? But so that's why I put random in quotes. But eventually it's random, and all possible combinations are made. All right. So now we've rearranged the DNA, and all that DNA in between here is removed from the chromosome and is actually deleted out. All right? Now, the consequence of this rearrangement at the DNA level is that the gene, these genes now become transcriptionally active. And the reason they become transcriptionally active is the following. Here's up front of each of these in the variable region gene up in the germline, you have a promoter that drives the expression of these genes. But the promoter is not a particularly good promoter, and you don't get transcription initiating from these things normally. It requires some help, and it requires the help of an enhancer. Now, I assume you had enhancers in biochemistry? All right, so it requires an enhancer in order to get good transcription initiating at that promoter. There is an enhancer in the system, but it's sitting here. Right there, right upstream of C kappa. And if you remember the properties of enhancers, they can enhance transcription from promoters. But one of the requirements is that they have to be within a certain distance of the promoter. Well, in the germline, this enhancer is sitting far away from any of these promoters, and so it can't enhance transcription, and that's why you don't get transcription of these genes. After the recombination event, however, this enhancer is now brought close enough to the promoter in the rearranged gene so that it now can initiate transcription from this promoter. It's still far enough away from all these other promoters it doesn't initiate transcription from those other promoters. It only initiates transcription from this, because this is the only one that's close enough now. So we now start transcribing this gene. And when we get the transcript starts here. We transcript the leader, the V, the J, and so on. You go here, and the end of transcription is at the end of the C kappa gene, and you make a primary transcript that looks like that. All right? This transcript then is processed, or spliced, whatever you want to call it. It's RNA processing, RNA splicing. And the processing is in such a way is that this intron is removed, and this whole area is removed. And we end up then with the messenger RNA for a kappa light chain. And the messenger now has all the coding sequences adjacent to each other. All right? This message then gets translated into protein. The leader segment, the leader region here, has got a signal, it contains a signal that says transport me to the lumen of the endoplasmic reticulum. So as this protein is being made, the leader directs the protein to go into the ER, and you end up with the protein in the ER. In the process of transport into the ER, the leader region is actually clipped off, and you end up in the lumen of the endoplasmic reticulum with a kappa light chain. What we had referred earlier to as the variable region is actually encoded for by the variable region gene as well as the J gene segment that was used. So the variable region is encoded by V and by J. What we had referred to as the constant region is encoded for by the constant region gene in the germline. Okay? And so now we have made a kappa light chain, and that's in the lumen of the ER. When this cell makes a heavy chain, then that'll assemble with the light chain. Okay? The heavy chain will assemble with that light chain, will make an immunoglobulin molecule, ship it off to the surface of the cell to be displayed on the surface of the cell. Okay? Any questions? All right, we'll stop here and pick up the heavy chain genes tomorrow.